Hi everyone, I'm Jacob Kraus. I'll be talking with you guys about phenotypic variation in Solanum carolinense populations within Japan. So first off, what are invasive species? Invasive species are species that are introduced outside of the geographic range that thrive and often outcompete the native species. The introduction can be intentional for ecological or financial means, such as crops or biological management strategies. There are currently believed to be over 5,000 invasive plant species which have been introduced to the United States alone. Of the invasive species seen throughout the world, many are introduced unintentionally via people. Trade routes and other modes of transportation lead to unintentional introduction of non-indigenous species across the world. So why are they important? These invasive species greatly impact the introduced range of the species. This is seen in the diversity of native plants within the introduced range. Over 80% of endangered species around the world are threatened or at risk due to the impact of these non-native species. The management of invasive species has posed a great problem around the world. Invasive species themselves pose a great risk to ecosystems and the economy. A study conducted by OTA in 2003 found that 79 invasive species cost approximately $97 billion in a span of 85 years between 1906 and 1991. Management control strategies devised to combat invasive species include biological, chemical, and mechanical control. So what are management techniques? Management techniques are methods used to control a specific species or several species. There are three main types of control methods: biological, mechanical, and chemical. Various management strategies can be utilized to manage or prevent invasive populations. So how are management techniques implemented? Biological control includes introduction of a natural predator in the introduced range to control pests. Mechanical control is applying physical damage to the species. This includes mowing, hoeing, etc. A picture of this can be seen on the bottom right of the slide. Chemical control is the prevention strategy, utilizing the spray or application of chemicals on the plants, as can be seen in the top right of the slide. The use of chemicals is widely used among farmers and is reported to cost $4 billion annually, with 3/4 of that as a result of invasive weeds. The United States spends over $100 million each year on aquatic weed control alone. In order to better understand the application of these control methods, we must better understand the study system. It is important when considering the implementation of management strategies to understand when we can or can't apply the same control method to populations of the same species. So if there is significant variation between populations of an invasive species of plant, it would be expected that the invasive populations would be dealt with utilizing different management techniques such as mowing, chemical, etc., as we talked about before. However, if the weeds were not significantly different from one another throughout their invasive range, then they could be handled or eradicated in the same manner. A lack of genetic variation would confirm a founder effect had taken place and help us better understand the evolutionary history of invasive species as a whole. So next, this is our study system. Our study uses a perennial weed, Solanum carolinense, also known as Carolina horse nettle, as the focal organism. S. carolinense is native to the southeastern United States, as can be seen on the map on the right. And more specifically, the plant is native to the Gulf Coast region in the United States and is spread as far north and westward as California and Canada. It's commonly seen in pastures, river terraces, and roadside plantings, and it grows rapidly in hot temperatures and is capable of replicating through seeds, pollinated by bumblebees and carpenter bees, as well as by vegetative root cuttings. It defends itself well in its native territory with solanine, trichomes, spines, and highly poisonous berries. S. carolinense was introduced in Japan within the last 100 years. The weed rapidly became invasive in the 1990s. For example, a study conducted by Hiroshima in 1989 found that although it caused many problems within fields and pastures, it was only present about 0.4% of the farms that were surveyed. 10 years later, another study found the weed present in about 25% of the farms that were surveyed. These studies support the idea that S. carolinense is a growing concern in its introduced environments. So why is Solanum carolinense a good model organism? S. carolinense would be an effective model organism for studying how phenotypic diversity poses challenges to the management strategies of invasive species. Many plants boost numerous defenses from spines and trichomes to chemicals that aid in the resistance against herbivory. Plant defenses can hinder the deployment of biocontrol agents. Therefore, understanding how plant defenses change in invasive species is useful in selecting the appropriate management strategy. So what type of defenses are utilized by Solanum carolinense? The image on the upper left shows a dried leaf of Solanum carolinense. These leaves have both mechanical and chemical defenses. The mechanical defenses act as a first line of defense and are the physical barriers that deter against predatory. There are two mechanical defenses utilized by S. carolinense, trichomes and spines. The image on the right shows a leaf of the nightshade under a microscope. The star-shaped spikes shown are the trichomes. The image on the bottom left is also an S. carolinense leaf under a microscope. Along the midline of the leaf, you can see spikes radiating outward. This is also indicated by the red circle. The spikes coming from the midline of the leaf are called spines. On the other hand, the nightshade has secondary defenses against predation that are comprised of chemical defenses. The chemical defenses of this species include alkaloids and proteinase inhibitors. So in order to better understand the invasive species that affect our community, we must look at invasive species on a broader scale. Our research is distinctly looking at the phenotypic and induced response variation among different populations. With a better understanding of the evolutionary history of the nightshade, we can answer the question, can we use universal management strategies for invasive species populations across Japan? So what do we predict? We hypothesize that the phenotypic diversity is low in Japanese populations of Solanum carolinense because clonal reproduction limits the genetic diversity among the plants. S. carolinense likely underwent a founder effect when introduced to Japan. So we predict from this that the morphological traits and induced responses between populations will not vary significantly from one another. We expect to see some variation in the plants within the study due to the plasticity of their characters, but we expect to see this throughout all plants regardless of population. So, to better understand whether or not different management strategies should be used for management of Solanum carolinense, we must first understand the distribution of the 7 surveyed populations. We will then be able to better understand the pilot data from a common greenhouse experiment in Forest Grove, Oregon, and a population survey from throughout Japan. Looking at the distribution of surveyed populations within Japan, the figure on the left comprises all 7 of the Japan populations that were studied in our pilot data and displays the geographic location where each population was observed. The table on the right displays the distance between populations in kilometers. The table and the map together indicate how distributed the sampling sites were from one another. For our preliminary data, we have analyzed data from 2 separate studies, an observational field study and a common greenhouse experiment done on S. carolinense in Japan and the United States, respectively. To begin the study, we set up a common greenhouse experiment on the Pacific University campus in Forest Grove, Oregon. We chose 3 populations within the seed bank that were collected by our advisor, Dr. Stacy Halpern, in 2018. From the populations, we randomly selected 10 maternal plants from which the seeds came from. We then planted 10 seeds from each maternal plant to yield a total of 100 seeds per population. These seeds were planted in a 50-cell planter on February 20th, 2020, and monitored for approximately 4 weeks. We surveyed the germination rate and time of the 3 Japanese S. carolinense populations that we picked, KTRA and KTRB, which are located near the Takano River of Kyoto, Japan, and TSI, which was just north of Tokyo. So what did we see with our greenhouse experiment? These are the results of our common greenhouse experiment. So from the collected data, we calculated the rate of germination as seen in the table on the right. The table indicates that the germination rate observed in our study was greater in KTRB compared to that of other populations. This is of unknown significance. The graph on the left, it shows the days until germination for each population on the y-axis and the site location on the x-axis. The median of the germination time for each site was not different among populations. We ran an ANOVA test on our results and determined the results expressed on the graph were not statistically significant with a p-value of 0.344. The lack of variation observed in generation time goes against our prediction. The lack of variation argues that the same management control methods could be used throughout the 3 populations that were surveyed. The population of S. carolinense along the Takano River was genetically researched, where evidence suggested that there was little to no genetic similarities within subpopulations, and a spread of S. carolinense across Japan was not likely due to seed reproduction. The study provides a genetic background of 2 populations which we used in our pilot data. We obtained our preliminary data for all measurements excluding germination rate and time from a study completed from July to August of 2018 by Dr. Stacy Halpern and colleagues. Plants from 7 populations across Japan were surveyed for physical traits, including plant length, total number of leaves, leaf damage, leaf area, specific leaf area, number of trichomes, and number of spines. We conducted an ANOVA test for each trait that we surveyed. We constructed box plots from the data. To understand the data, we must first understand the box plot. A box plot breaks the data into 4 parts, 2 boxes and 2 whiskers. The boxes represent the central 50% of the values divided by a dark line, which is the median. So the central 50% of the values is also referred to as the interquartile range. So while the whiskers represent the 25% of the data that falls outside the interquartile range on each side of the boxes. Why do we use box plots and why are they important? A box plot allows us to have a better indication of how the values in the data set are spread out. It is important to note the distribution of the data as it relates to the variation among traits that we are looking at in this study. The 2 leaf traits we surveyed were specific leaf area and leaf area. These traits are on the y-axis on their respective graphs. Throughout our analysis, we placed the traits on the y-axis and put the sampling sites on the x-axis. The leaf trait graphs indicate variation between each site, but also indicate consistency in the trend of each population. We see this consistency between graphs with how the box plots look compared to one another. Take the median values, for example. If we are to see consistency, then we'll see the medians being in a similar position compared to each other. We also see that the interquartile range does not differ significantly between graphs. This is expected as the 2 traits are related to one another. All the results from this study, including the 2 graphs you see above, are statistically significant. We looked at 3 different defense traits between the 7 populations. Average leaf damage can be observed on box plot A, and average trichome and total spines can be seen in box plots B and C respectively. There is significant variation between sites, but the variation was not consistent among traits. The variation among traits can be best understood comparing the trend of each box plot. The interquartile range of HSS on the far left in figure B is small compared to the other populations, while the interquartile range is vast when looking at figure C for the same population. This goes along with our prediction that there is great variation among traits, and therefore different management strategies would need to be implemented to control this invasive species. The surveyed size traits were total length of the plant and total number of leaves. The total length of the plant can be observed in box plot A. The total number of leaves can be observed in box plot B. We can see the same overall trend among both graphs, indicating that there is consistency of variation among the traits. We also observed variation among the sites. This was expected as the 2 traits are related to one another. So what can we conclude from our pilot data and where do we go from here? So this brings us back to our main question. Can the same management strategies be utilized for different populations of Solanum carolinense? Our pilot study helps to provide us with a solid understanding of this question, but does not help us to conclude anything from the results. To better understand the results from the pilot data, further research will need to be conducted. A common greenhouse experiment could be used to help us determine the best course of action in managing the invasive species throughout Japan. So with this further research, we will look to answer 3 main questions. 1, how much do populations vary? 2, is there an effect of the management strategy? 3, does the management effect vary among populations? So let's look at our methods. Our proposed common greenhouse experimental design. We will survey the 7 populations used in the previous study, as well as shoot clippings from the same maternal plants. The populations will be grown in a greenhouse for 4 months, and the mature 4-month-old plants will be randomly assigned to 5 different treatments, as well as a control group for each greenhouse. There will be a greenhouse designated to each control method, and within each greenhouse, different control methods will be used. For mechanical control, we'll use a specific mowing height. For chemical control, we'll have to apply 2 of the most commonly used herbicides, which are glyphosate and 2,4-D. For biological control, we will treat the plants with Manduca sexta and Frumenta nudinella. Plants will be randomly cross-pollinated with individuals of the same treatment once they flower, and we will do this by utilizing bombus bee colonies to achieve the random pollination. Measurements of the fruit number, dry biomass, mortality rates, and ramette density will be taken after 8 months, and the results will be analyzed using a two-way ANOVA test to determine the significance of our findings. So we created expected results from this, and this is what we thought we would see. Our mock data shows that we expected to see high variation among the populations. This is due to both population site, treatment, and phenotypic plasticity. The performance traits we analyzed were significantly affected by both the treatment group type and the population site. There was diversity when comparing populations to each other, as well as when comparing treatment types to one another. This goes with and against our hypothesis. We expected to see variation among treatments, as this indicates the ability of the control method on the plant. We did not, however, hypothesize that there would be as much variation among populations, indicating that there is potentially another variable that was not considered. If you look at RAN, the third population from the right on both box plots, you will notice that in the control group, it has a high fruit count compared to the other populations. While in the mowing group, the same population has one of the lowest fruit counts. This is a significant decrease. On the other hand, we'll see a less noticeable shift in KTR2, the third from the left on both graphs. The interquartile range remains about the same, but the distribution between the control and mowing box plots was altered. The median is approximately the same, but there is a slight shift downward for the interquartile range. We can expect from these results that there is an effect caused by the management strategy, and the management effect varies among populations. So, in conclusion, from the preliminary analysis and pilot data, we can conclude that there was high variation among sites. Because of high variation, we would expect different management strategies to be necessary to control the invasive species. But there could be another variable that could account for the high variation we observed in the results that we were not accounting for. Our results from the pilot study and our expected results suggest that the different management strategies must be applied to the different populations, arguing against a founder effect. Our proposed methods will compare traits within populations as well as between populations. We'll be able to better determine the phenotypic similarities from these populations and determine if their traits are similar or significantly different. The results in the study will give us a better background and provide us with enough information to determine if similar or different management strategies should be applied to Solanum carolinense populations in Japan. All right, that is the end of my presentation. Here is my bibliography. Thank you for listening to my talk, and have a great rest of your day.